This video is all about the streak plate technique, which is an isolation technique. So essentially what we're going to do is introduce bacteria onto an agar plate. We're going to streak that sample of bacteria across an agar plate with the end goal of having isolated colonies. So let's take a look at how we're going to do this and really why we're going to do this. Let's start there. So this is an isolation technique, and so we're trying to isolate individual species that are present in a mixed culture. And so when we go out and take water samples, for instance, there are tons of different bacteria in there. And by streaking them on an agar plate and looking at how they grow on an agar plate, we can identify what microbes are in that water sample. If we have an unknown organism, we need to get individual colonies before we can do further testing on it. Because remember, a colony is a mass of identical cells. So with this technique, we're going to be introducing individual cells across the plate. And when we put it in the incubator, those single cells will divide over and over and over again. So by the time we come back into the lab 24 hours later, that single cell has divided into millions of cells, all in a little cluster that we can see with the naked eye, and that is called a colony. And all the cells in that colony are genetically identical to one another. So we can use part of a colony when doing testing because we know all the cells are the same. Okay, so they're all going to behave the same. And so that's how we study bacteria. So with this technique, you're going to be streaking a sample across a plate. Sounds simple enough, but there's a little bit more to it. And that's because when you originally dip your loop into a broth culture, for instance, you are picking up thousands and thousands of cells. When you initially touch that loop to your plate, you're depositing thousands and thousands of cells onto that agar plate. But remember, the end goal is to have individual single cells. So we're going to have to streak that plate, because the more you streak a plate, the more you're spreading out those cells. And so we're going to have to actually have a more organized approach to our streaking. We can't just go all willy-nilly, so that by the time we're done with this technique, again, isolated colonies. Now, one reason this technique is so important is not only for identifying what species are present, but it can actually tell us whether or not we have a pure culture or a mixed culture. Because the problem with broth cultures, the liquid cultures, is that if, let's say, our tube is supposed to only have E. coli, well, when that E. coli grows in the broth turns cloudy, right? Science term is turbid, and that's great. We know there's bacteria in there. But if our culture was contaminated, so there was E. coli, but there was also salmonella, it would still look turbid. We can't tell just by looking at a turbid tube whether or not it's just E. coli or it's E. coli and something else. So that's the big drawback to broth cultures, is you cannot tell whether or not something is contaminated. So after doing this technique called the streak plate, we also refer to it as the quadrant method, because as you see here, we're going to be dividing the plate into 4 sections or quadrants. And so after we do that, right, we're going to have those isolated cells, isolated colonies, like you see here on this picture. But notice what else you see on this particular picture. Do you see these red colonies? Then there are also beige colonies. Well, the beige colonies are all one type of organism, maybe E. coli. But these red colonies are a different organism. It's actually an organism called Serratia. And so what we can do is we can look at the colonies that grow on an agar plate and figure out how many different species we have. And once you've been doing this long enough, you figure out just by looking at them what this species is. And if we don't know what this species is, we can do testing to figure it out. So that's another benefit of doing this streak plate technique is you can figure out if your culture is contaminated or not. Pretty snazzy. All right, so let's talk more about the details of this actual technique. So whenever we are going to do any type of inoculation or any type of technique, you always label your media. And so we are always going to label the bottom of an agar plate because the lids can come off, right? So if everybody has their lids off, there's the potential that they'll get mixed up and you won't know, which lid belongs to which plate. So you always label the bottom of the plate. And so you're going to divide your plate into 4 sections. And honestly, both of these approaches are perfectly fine. I do the right-hand side personally, but a lot of my colleagues do the left-hand side. Either works, okay? And you can label any quadrant 1, 2, 3, 4. You just have to kind of go, in a circular motion. But this is the way that I label them. So you've got your 4 sections. And what we're going to do is using aseptic technique, you are going to get bacteria on your loop. And then what you're going to do is you are going to streak that loop across that first section. Okay. And what I want you to notice here is that there are as many streaks as possible. You try not to overlap your streaks. You get as many as possible, but you streak from edge to edge. Okay. Because remember, the goal is to spread out these cells as much as you possibly can. And so you're going to want to cover as much of this space as you can. So the problem with the picture on the right is they haven't really put any cells on the side, and that's actually going to be pretty problematic here in just a second. So let's look at the next slide. So after streaking in quadrant 1, you tend to rotate your plate because it's always easier to just streak the top section of the plate. So you rotate your plate so that the sterile section 2 is now at the top. And you're going to flame your loop. That's important because you want to get all the bacteria that's currently on your loop off of it, because we're trying to go from thousands and thousands of cells that you put in section 1 down to 1 cell, right? When we're all done. So by flaming your loop, you kill everything, but you've got to put something in quadrant 2. So notice what's happening here on the left. Do you see how some of the streaks go into quadrant 1 because they're stealing cells? Spread them across part of quadrant 2, steal a few more from section 1, spread them out in 2. Okay, and you're just going to kind of do that a little bit. So you remember, you have to steal from the previous quadrant to get cells on your loop. All right, now notice what's wrong here in the right-hand picture. Do you see how after flaming their loop, after they finished quadrant 1, when they came over to quadrant 2, they didn't steal any cells? That means they're moving their loop across the plate, but there's nothing on their loop. So there's not going to be any growth there because they didn't introduce any cells to that section. Okay, so once you have completed section 2 or quadrant 2, you're going to flame your loop. You will flame your loop after every single section. Oops, sorry. So again, you rotate your plate because again, it's easier to streak at the top. So you flamed your loop after completing quadrant 2. So you're going to set your loop in quadrant 3. You're going to steal from 2, streak it out, steal maybe a little bit more from section 2, streak it out, and so forth. Once you're done with section 3, you're going to flame your loop again, rotate that plate, and now this is the last quadrant. So you want to get as many streaks as you can. So they flamed after quadrant 3. Now they're going to steal from 3, spread it into 4, steal from 3, spread it into 4. But then eventually they're going to get as many streaks as they can in here in quadrant 4. Okay? Because ideally at the end here, you're down to individual cells. And then you're going to flame your loop and you're all done. But I want you to take a second and look at what's happening here. Notice, remember, we started in quadrant 1. So we had taken cells from our broth tube and we spread them out. At that point, once you're done with section 1, you put your broth off to the side. You never go back into it, okay? So we flamed our loop. For section 2, we stole from section 1, but notice these streaks, y'all, in section 2. Do you see how we try not to overlap? We also spread it out as much as we can end to end to ensure that we'll have cells to steal in section 3. Once we flame our loop, notice we never go back to section 1, okay? Because we don't want to grab all those cells. We're trying to get fewer and fewer cells. So you just want to be careful that you don't overlap your streaks and you don't accidentally go back into any of the other sections. And that's really a concern for quadrant 4, because if you're not paying attention, it's easy to accidentally dip back into quadrant 1 or accidentally go too far and get into quadrant 2. In which case, you're just introducing lots of cells back into that quadrant where you only want a few. Okay, so let's take a look real quick. So we just looked at this picture on the far left. That was the good one. Notice here in the middle, do you see what happened in quadrant 4? They dipped back into quadrant 1. There are thousands of cells in quadrant 1, y'all. So now suddenly you're picking up thousands of cells every time you dip back in. So you won't get isolated colonies. But notice on this far right-hand picture, there's a different problem. They only stole from quadrant 3 twice, but then they did this little really wimpy streak here, okay? So even though you have less cells in quadrant 2, even fewer cells in quadrant 3, you're still wanting to spread them out to ensure that you get isolated cells and therefore isolated colonies. So you don't want to do this little wimpy streaking that you see here. You want to just really take all the space that you have. All right, so we are not going to watch this video, so I'm going to leave that to you. So this is a pretty good streak plate here. Notice in this fourth and final quadrant, there are plenty of isolated colonies. Technically, an isolated colony is when a colony is by itself enough that you can confidently grab cells from just that colony and not worry about grabbing anything else. So this cluster down here is not isolated because you can't confidently grab cells from just any of them. They've merged together, right? So you see several isolated colonies here in quadrant 4, which is exactly what you want. Here are a couple other pictures that just show you nice, pretty isolated colonies. And I do want to point out, like if you look at this right-hand picture, you'll notice there's 1 section, 2, 3, 4, right? And then it does come all the way down here. So if this person had not done as many streaks as they did, you wouldn't have gotten isolated colonies, okay? Because they really didn't, they really stole a lot of cells each time, way more than necessary. When I do this technique, I only steal 2 or 3 times, not 20 like you see here. But they did get isolated cells, and that's the goal. So for these, I want you to pause these videos and look and see what went wrong. I'm not going to give you all the answers, but I will give you things to think about. So look at for this picture. They only have cells in their first section. Okay, so there's a couple things that could have happened here. What do you think they might be? Had they drawn their quadrants, it'd be a little easier to see. But notice they didn't go to the edge of the plate all the way. Another thing that may have happened is when you flame your loop in between quadrants, you have to let it cool down, right? It's just like we can't go walk into a blazing fire. We'll burn up. If you don't let your loop cool before you try to steal cells, you'll just burn those cells. And then you won't have anything that you're actually spreading across the plate. So those are 2 possibilities of what could have gone wrong. Check out this one. What do you think happened? So you have to kind of look right here, section 1, 2, 3, and 4. Notice how many times they dipped back in, right? You only need to steal from the previous quadrant a couple times, and then you really just want to streak it out so that you're decreasing the number of cells you have in that quadrant. So it's very easy to be paranoid when you're first learning this and you want to steal because you want to make sure you're actually grabbing cells. but too much stealing can also be problematic. All right, look at this plate. So they did a good job with quadrants 1 and 2 and 3. You can tell there's fewer cells each time. You can start to see some cells by themselves over here. Sorry, my mouse is on the wrong screen. All right, so lots of cells, still quite a bit, starting to thin out. So a good job here, but notice they dip back into that first quadrant. Okay, so they were doing a great job decreasing how many cells they had, and then they picked up a bunch more. All right, last but not least, for the streak plate technique, take a look at this. What do you think went wrong? So you can tell, you can see here these streak marks. So they tried to steal, but notice there's empty cells, or I'm sorry, there's no cells here or here. So odds are when they went to steal, they just didn't grab much of anything. Okay, so they tried to streak it out, but because they didn't do enough streaks in this second quadrant, there wasn't enough to steal and spread. All right, y'all, that is the end of this video.